In this video, we will discuss about this chimeric antigen receptor construct or the CAR construct, which is generally used either in the T cells or in the NK cells for this immunogenic therapies. And there are some minor difference between the CAR construct in the T that is used in the T cells and the CAR construct that are used in the NK cells. And before going into that, we will first understand different kind of CAR construct. And we will start with this first generation CAR constructs. And each CAR constructs will have this extracellular domain and this hinge to connect the extracellular domains, and this transmembrane domain and the intercellular domains that is required for the activations of the signals, and that determines how strong the activation signals will be generated inside the T cells. And in the second generation CAR construct, they have one additional co-stimulatory domains. And in the third CAR construct, they have two co-stimulatory domains, and it could be either this 4-IBB or CD28 for the T cells. In the fourth generations CAR construct, they have both the co-stimulatory domains along with a payload. There is a payload also been attached. And now we will understand some of the difference between the T cells CAR construct and the NK cells CAR construct. The T cells CAR construct, they usually use this 4-BB or 4-IBB and CD28 along with this CD3. And whereas these things also works for NK CAR construct, but these are not naturally expressed in the NK cells. So the natural choice for the NK cells is this NKG 2D, DAP10 or DAP12 instead of this CD3, CD28 and 4-BB. So this will be the natural choice, but what it will does is it will make this NK cell CAR construct hyperactive, and we have to check its activity, whether it will cause any other undesired effect or not. And how this CAR T cells works is shown in this figure on the bottom side, where the tumor cells, since the CAR T cells in the case of this extracellular domains, they contains the receptors that can detect the antigens, tumor antigens or TAA, tumor associated antigens, and thereby the intercellular signals get activated and it will release the cytokines. Cytokines are mainly released because of the co-stimulatory domains such as Granzyme, perforin, that will trigger apoptosis of the tumor cells. And here the short version is shown where only the extracellular domains or these receptors interact with the antigen. But in reality, along with these receptors, antigen receptors, there are other things also needs to be activated, such as FASL pathways, trail pathways, and the CD16 pathways that will activates this T cells, CAR T cells or CAR NK cells, and that will lead to the release of this cytokines such as granzyme, perforin, that will lead to the apoptosis of the tumor cells, and this apoptosis will cause release in the antigens in the cellular matrix, which will then be recognized by the dendritic cells, and the dendritic cells will then help in the activation of T cells and the B cells, and thereby accelerating, and these T cells and B cells will release. B cells will generate antibodies that are specific to those antigens that are present on the tumor cells, and T cells will release cytokines such as IFN, gamma, TNF alpha, interleukin 12, and thereby accelerating the immune response against the tumor cells. So this is in brief the how is construct of T cells and CAR construct for T cells and is differ from the CAR construct from NK cells, but is in many cases people have shown that this same CAR construct for the T cells can work for the NK cells, and we have also understand the basic phenomena how this CAR T cells helps in the activation of the immune system of the host. That's all for this video. If you need to understand more about this chimeric antigen receptors construct, you can go through the reference that has been provided in the description. Thank you for your time.